Hello and welcome to Digital Medboard. Here we go. The order of draw for collecting blood samples is based on how sensitive each test is to contamination. Following the correct sequence is crucial to avoid inaccurate results, especially when additives from one tube may interfere with others. The first tube to be filled is the blood culture bottle, as it is highly sensitive to contaminations. If you collect blood in the other tubes first and then transfer it to the culture bottle, the sample may already be contaminated. Therefore, collect the sample first in the culture bottle to maintain sterility. Next, collect blood for coagulation assays such as prothrombin time, activated partial thromboplastin, fibrinogen assays, and factor assays. These tests are highly sensitive and can be affected by contamination from other anticoagulants. So, the blue top tube contains 3.2% sodium citrate should be drawn second. The best way to remember the upcoming tubes by traffic light sequence. Think of the order like the colors of a traffic light, followed by additional tubes. Number 3. Red top tube, which is a clot activator tube used for serum collection without gel. This tube contains silica, a clot activator. Number 4. Yellow top tube contains gel and clot activator. It helps create a barrier between the serum and cells during centrifugation. Number 5. Green top heparin tube, used for stat biochemical and genetic testing, which contains heparin, an anticoagulant. Number 6. Lavender top EDTA tube, used for complete blood count CBC, HbA1c glycated hemoglobin, and tests requiring prevention of degrading like PTH, ACTH, ammonia. EDTA chelates calcium and inhibits proteases. Number 7. Gray top fluoride tube, which collected at last. The gray top tube contains sodium fluoride and potassium oxalate. It is used for glucose estimation and is known to interfere with many other tests. Hence, it is drawn at the end to prevent contamination. If you found this video helpful, don't forget to like, share, and subscribe to Digital Medboard. Got questions? Drop them in the comments below. See you in the next video. Until then, stay tuned. Choose.